Hi, my name is Bailey Foster, and I am in Dr. Keith Paterka's EES 100 class. My presentation topic today will be soil composition comparisons using XRD and XRF data. A little background on XRD and XRF stuff for those who aren't familiar with these processes. We run samples in the X-ray diffraction to analyze crystalline structures to reveal chemical compositions. X-ray fluorescence is a quantitative analysis showing us the elemental composition, and it gives us a percentage of how much of a material is present. All right, so motivations and goals. What are we doing? For this project, we took XRD and XRF readings of various soil samples that we've collected either from different areas or different depths. I did a mix of both, taking samples from my family members' houses from both the surface level and a couple inches deep. Why are we doing this? We're doing this for a few reasons. We are interested in seeing what makes up our soils and if there's anything abnormal present. Personally, I was interested in seeing if there was a difference in soil compositions across different parts of town, from East Clovis to West Fresno. And lastly, we are familiarizing ourselves with the machines and learning how to interpret the data. Knowing how to use these machines and explain the data to others is a valuable skill as a geologist. Our data and methods. So for this project, I gathered 5 dirt samples from select locations and depths, which will be explored more in the next slide. Before sending the samples to be run through the XRF and XRD, I first had to dry and grind them up to a superfine powder. Some of my samples were drier than others, but I still stuck them in the oven to suck out any residual moisture. The process for getting the samples in the XRD aren't that complicated. The soil is dried, ground up, and gently pressed into molds. On the other hand, to make the XRF pucks, we weighed out 10 grams of the ground up soil and 1 gram of a binding powder. After mixing those two thoroughly, we poured the mixture into a mold, put that in an automatic press, which pushes down all that mixture to approximately 15 tons. That puck goes into a sample holder and then into the machine for it to be read. The samples stayed in the XRD for about 3.5 minutes, while the pucks stayed in the XRF for about 30 minutes. Back one. Okay, these are all the raw samples that I picked up around town. Sample 1, there we go. Okay, sample 1 is from my backyard in Northeast Clovis at the surface level. My backyard is pretty dry with very little plant matter, and the soil is pretty fine with some larger grains thrown in there. Sample 2 is from my mom's backyard in Northwest Fresno at the surface level. Her soil is very fine, almost like a clay. Sample 3 came from my brother's house on the border of Fresno and Clovis at around a foot deep. That soil is very coarse, almost like sand. Excuse me. Sample 4 is a deeper sample of my mom's soil, taken at around 6 inches below the surface. And sample 5 is another deeper sample of the soil in my backyard at around 6 inches below the surface. All right, and this is the XRD graph for sample 1. Each peak corresponds with a specific compound. Quartz, for example, is the largest peak displayed. The higher the peak, the higher the concentration. So looking at this graph, there's a lot more quartz in my soil than dickite, which is a type of clay. A substance called zinco-woodwardite was found, which is a sulfate, which is typically a naturally occurring mineral. And this is the XRF data for sample 1. It gives us the quantitative data showing how much of each substance is present. Comparing this data to the XRD graph, that large quartz peak corresponds to the large percentage of quartz on this XRF sheet. Aluminum oxide is also present as well, which is found in a lot of clay. And this is soil sample 2's XRD graph. which, similar to the last XRD, quartz has a very high peak, with albite, plagioclase feldspar mineral having the second highest peak. Riebeckite down here is a sodium-rich silicate mineral, which is present as well, which is shown by the third largest peak. Dickite, though not abundant, is still present down here. And this is the XRF data. Shows that quartz once again makes up most of the soil with some aluminum oxide. Again, the largest peak corresponds to how quartz comprises most of the dirt at about 70%. There's a lot of cobalt in here as well, which may be a potential essential nutrient for plants. There was a garden in my mom's backyard over the summer. Maybe some of the fertilizers that she used could have seeped into that soil. And unlike the last two graphs, albite has the largest peak on this XRD graph. And manganocumingtonite, a mineral that I wasn't expecting to find, has a pretty tall peak. It's a type of silicate with some manganese attached to it. And this is the XRF data. With albite being more prevalent in the soil, there's going to be more CaO present in the XRF scan than the last two. The manganocumingtonite being present also correlates with the increase in MgO. Quartz is still the majority of the sample because oxygen is the most abundant element and silicon is the second most abundant element. All right, and quartz is the largest peak on sample 4's XRD graph, and albite is the second tallest. Dickite is present, which makes sense because this is a deeper sample from my mom's backyard, which also contains dickite. Kaninkite, a phosphate, is found as well, that peak. Phosphorus is another one of those minerals found in fertilizers, so maybe more of that fertilizer that was used seeped into the soil. And once again, quartz makes up about 70% of the sample, which corresponds to the large peak it has on the XRD graph. Cobalt is relatively high, as well as barium. Barium is a non-essential mineral to plants and may have reached the soil after getting into the groundwater. And on this graph, both quartz and albite have significant peaks, which corresponds to sample 1's graph, as this is a deeper sample of that same soil. Kaninkite is once again present, showing the existence of phosphorus. Fluorescence, this is down here, is a mineral that once again I wasn't expecting, and it's of the mica group. So fluorescence contains some iron, which explains the higher percentage of iron 3 oxide compared to the other samples. The iron oxide levels here in the deeper sample are very similar to the surface sample. Like all the other XRF data sheets, quartz makes up the majority of the soil. So in conclusion, after taking samples out of my family members' backyards, I found really nothing out of the ordinary. There were some minerals that I didn't recognize and had to do some research on, but a lot of the compounds present stayed relatively the same from sample to sample. If I were to continue this research of the composition of soils, I would take longer scans of each sample rather than a 3-minute scan in the XRD, and take samples from a wider range. Taking samples from Fresno and Clovis and somewhere in between showed that there weren't a lot of differences between the soils of those areas. Thank you for your time, and I hope you have a great rest of your day.